All right. Well, I'd like to thank Jill and the organizers for inviting me to share this work on this course that I've developed. This is the first time I'm speaking about this work, so I'm really excited to get to present this and hear all of your feedback. So just for a little bit more of an introduction, so I am let's say an RNA biophysical chemist. My background is in RNA dynamics and RNA protein interactions, and our research group studies RNA structure-function relationships and ligand recognition. And so when I started as an assistant professor in 2019 in the chemistry department at the University of Nebraska-Lincoln, the department was rapidly growing in the chemical biology field. And to meet this growing need, the department needed to have a laboratory course in chemical biology. And so I was really excited to take on that challenge and develop this chemical biology laboratory course. So I want to start by just asking this question. Please feel free to type this into the chat. But when you hear someone say, I took a chemistry lab course, or just the words chemistry lab, what are some things, like what are some images or phrases, words that come to mind?
So in the chat, people said beakers and test tubes, stinky chemicals, test tubes and flasks. That's what they think of.
Okay, thanks. These are the types of things that come to mind, right? If you're thinking of inorganic chemistry, colored solutions, lots of chemical formulas, some organic molecules, beakers, flasks, hopefully not too much smoking or solutions, but these are the types of things people normally think about. But I really want to kind of challenge you and other people right in our department as I'm developing this course to think beyond kind of this silo of chemistry and expand beyond into biology, and more specifically chemical biology, right? So chemical biology is a relatively new field that was, I think, pioneered by Caroline Bertozzi and others, of using chemistry and organic synthesis and chemical principles to apply towards understanding biology. And so to develop this lab course, I wanted to incorporate a few different elements, both of the classical definition of chemical biology, as well as my own expertise in nucleic acid structure-function relationships. And so, of course, I wanted to include some synthesis elements, as well as biopolymers, such as nucleic acids or proteins, and ligand recognition. So in addition to incorporating these conceptual elements, I also wanted students to gain skills in how to generate hypotheses, how to interpret data, like real data coming from research and not cookie cutter experiments that have been done hundreds of times in other labs. To compare and connect different types of data. So for example, gel data with NMR data, and also to gain skills in scientific communication, because that's an increasingly important skill set that students need to be successful outside of college, right, into the workforce. And so some logistics about the course design. So at UNL, we have a 15-week semester. So this is the traditional one semester course. It was designed for upper level undergraduates. So Mark did a really great job of discussing the challenges with the introductory versus upper level coursework and the concepts that students tend to come in with particular sets of knowledge. So it was really designed for students who've already taken some introductory organic chemistry, some introductory biochem, so they know what DNA, RNA, as well as first-year graduate students. So in our department, several grad students every year come in with strong chemistry knowledge, but they're going into joining chemical biology groups and maybe need a little bit more shoring up of knowledge in molecular biology and more like biochemistry concepts. And so this class is designed to meet those needs as well within the department. In terms of assessments, we have pre and post labs, a lot of interactive websites and other things that students are working with, end of module presentations. So we have a presentation at the end of each of the 4 modules that I'll be talking about, and a lot of discussion and reflection. And at the end of the semester, there's a final presentation summarizing everything, along with a new research proposal where students present a like 1 to 2 year project of where they would want to take this project into new directions. And so I've taught this course twice so far. Once was in the spring of 2021, which you may imagine had some challenges. And so it was piloted first in 2021, and then it was most recently taught in the spring of 2023. So I just finished the second iteration of this course. And most of what I'll be sharing today is based off of the course that was taught in spring 2023. And so I'm going to summarize and kind of go over the entire course, time permitting, and also focus more on how we used Eterna specifically to shore up the RNA folding aspect. All right, so as I was thinking about these elements of synthesis for small molecules, biopolymers, and ligand recognition, I immediately thought of aptamers. And many of you are likely familiar with aptamers. I worked on the pre-cuisine riboswitch aptamer in my graduate work. So aptamers are biopolymers. They can be proteins, DNA, RNA, but they're polymers that bind a target molecule with high specificity. And there are natural examples of aptamers, such as bacterial riboswitch aptamers. So Mark did a great job of setting up the ribosome here, but in bacteria, riboswitches are regulatory elements that can regulate either transcription or translation by having a aptamer domain here and an expression platform downstream. And in this particular situation, the ribosomal binding site, where the ribosome binds to initiate translation, is sequestered in a stem. And so translation is turned off in this particular secondary structure. However, when ligand binds to the aptamer domain, it induces a change in the secondary structure to allow for accessibility of the ribosome binding site and translation initiation. And so in this way, through ligand binding, there can be a change in gene expression. There's also synthetic aptamers. So actually synthetic aptamers were discovered or developed first, even before the natural aptamers were discovered. And this work was really pioneered by Jack Shostak and many others in the 80s and 90s. And so aptamers in the lab, synthetic aptamers, are generated through selection methods such as selects or directed evolution. I don't have time to go into how that takes place, but the example I'm showing here is of the spinach aptamer, which is a member of the class of light-up aptamers that I'll be talking about for the rest of my talk. And so these light-up aptamers bind to typically metals, in addition to this cognate fluorogen, which is a small organic dye that's not normally fluorescent on its own, but when it binds to the RNA, there are conformational changes in the RNA that take place that help to limit the conformational freedom of the dye and allow it to be highly planar and highly fluorescent. And so this pair of the aptamer and the fluorogen together is a highly fluorescent complex. And so in 2019, when I began as an assistant professor, there's a paper that came out that reported this new class, or this new type of light-up aptamer called Pepper. And so in our group, we got really interested in it because it seemed to have a different type of structure that was a non-G quadruplex structure. For those of you who are familiar with light-up aptamers, many of them fold into G quadruplexes, which can have some challenges in biological applications. And so we were interested in this particular aptamer because it did not seem to form a G quadruplex. And it was shown in this paper in 2019 that the symmetric loop and asymmetric bulge were both required for ligand binding. And so it's a bit of a puzzle, like how can both of these pieces be involved in ligand recognition? And so I wanted to use this particular system for the chemical biology lab course. And so we had in our group developed what we call a mini Pepper, which is a little bit smaller, more tractable for the experiments that we were doing in our lab, primarily NMR spectroscopy. And so Pepper binds to this cognate ligand called HBC. I'm not going to go into what the HBC acronym stands for, but 530 refers to the emission wavelength when this compound is fluorescent at 530 nanometers. And so the dye by itself is not fluorescent, it's a sort of orange color. But together, when the RNA is added to the dye, it becomes this bright green solution. So it's a lot of fun to work with, really fun for students. And in 2021, the structure of this complex was determined by Aiming Ren's group, which beautifully showed how this RNA can bind to this small molecule through basically this bulge here coming up and interacting with the loop and with the small molecule. And this is really stabilized by several divalent cations shown in these gray spheres. But when I taught the first iteration of the course, the structure had not yet been solved. And so there really wasn't any structural information to go off of. All right, and so the main question that we had both in my research group and for this lab course was, how does the Pepper aptamer bind selectively to this HBC ligand, right? What are the mechanisms of ligand recognition here? And so to integrate these three components, I wanted to have kind of four main modules, right? So first, students would generate a specific hypothesis about an important residue for that interaction between the Pepper RNA and the HBC dye. They would then synthesize and characterize the HBC dye, synthesize and characterize the RNA, and then they would test this hypothesis in a plate reader assay. So hitting on all three of these domains that I wanted to explore in this lab class. And so I'm going to go through each of those modules. So first, in the first module, right, there's a pretty heavy lift. A lot of students coming into the class know very little about RNA. Maybe they know about the 4 bases and canonical base pairing, but not so much about hairpin motif, loop motifs, etc. And so some of the goals for this module was to learn the jargon and the basic rules of RNA folding. We had a PyMol tutorial to learn about 3D structure. So identifying interactions between Pepper RNA and HBC ligand, either through biochemical evidence reported in the literature or through these modeling activities. And then to design a mutant construct, making a hypothesis that this particular residue is important for ligand binding. I'm going to make this mutation, and I hypothesize that it will either increase or reduce fluorescence and binding. And then at the end of each module, we have this presentation session and discussion. All right, so this is really where Eterna really helped us a lot, because like I mentioned, this was a really new area for a lot of students. And so we relied heavily on the NOVA labs from PBS, where Eterna worked a few years ago with PBS to develop this RNA virtual lab. And one of the reasons, and we had previously used this quite a bit in our outreach efforts for middle school and high school students, and it seemed to be really successful. So I thought, this is going to work really well for college and graduate students. And so if you haven't played this virtual lab, it's a lot of fun. I really recommend it. And I know now Eterna has a new version, like within Eterna, for these types of activities. But it's a really kind of nice fundamental way to introduce students into RNA folding and function. So it covers the basics and then introduces different functional aspects of RNA with protein synthesis, the RNA world hypothesis, and viruses. And so in addition to the PBS NOVA labs, I also had students do this puzzle on light up switches. So this was a really wonderful puzzle that had previously existed on the malachite green aptamer. This was the first aptamer, a fluorescent aptamer that had been developed in 2003. And so I had students just do this round one. And so the challenge here for this puzzle is that students need to design one sequence of RNA that can fold into 2 different secondary structures, but only one can bind the malachite green ligand. And so students really struggled with this. This was really, a challenge, but every student got it and they actually really, really enjoyed it. And so this is one example solution from a student. And I'm highlighting here the aptamer region where the ligand binds into this symmetric loop in one state. And then in another state, there's a rearrangement of the base pairs where the ligand can no longer bind. And I will also mention here that a student in our research group, Mamadou Camara, will be talking tomorrow about RNA structural dynamics. And so if this is intriguing to you, I recommend you go to his talk. It's going to be really awesome. All right, so some reflection. So on a post-lab assessment, I asked students to share their thoughts about this, and I really was really impressed by a lot of the student comments. So here, this particular student had this assumption or misconception that the same sequence would always mean the same secondary structure. But what they realized was that the conditions, right, whether ligands were present, would really affect the structure. This student really liked the puzzle, helped them to understand nomenclature, and it helped them to really understand how dramatic these structural changes can be, as well as the conditions. And this student was starting to formulate some rules, right? They were starting to think about what are some rules for folding and where could base pairs be placed in a stem to have different types of outputs, right? So here that, the weakest point is at the end of stems. And so by placing a GC at the ends, that can stabilize those stems. AU bonds, they were starting to think, oh, maybe AU bonds are good to put in the middle of a stem. UG bonds are great for, if you want some instability to break bonds and change shape. So I thought this was really interesting to start to gain some insights into how students are thinking about these fundamental aspects of RNA folding through this Eterna puzzle. All right, so then getting into module 2, this was focused on synthesis of the HBC, right? So because this is a chemistry in a chemistry department, I wanted to have some, fundamental organic synthesis to get that kind of traditional chemical biology aspect. And so the goals here were to apply and expand organic chemistry knowledge. This particular reaction is really nice. It's a one-step, one-pot reaction. It's a variation of the aldol condensation. It's a really fun name, the Canova-Nagel reaction. So students really enjoy learning about this and how they can apply their existing knowledge of aldol condensations into use of additional catalysts to have some variations. And also we apply many methods of characterization to evaluate purity. So we bring in NMR, mass spec, and HPLC. And so students need to evaluate all these different types of data to validate that they have the compound they think they have and evaluate the purity. And so for a little glimpse into that, it's a really fun reaction, changes color from a lighter orange at the beginning to this darker orange. This is a very slow reaction. It takes about 4 days and it ends up with a precipitant. And it's relatively straightforward to purify. We do on column purification, which is not really necessary because it's already pretty pure. But students have a lot of fun recording videos. You can see here's the product in this darker orange. And students analyze their NMR data. One student had this really beautiful color-coded guide to assign the identity of the resonances in this 1D proton NMR experiment. And then moving quickly to module 3. So this is focused on RNA synthesis and characterization. And so here I really wanted to add some elements of core molecular biology concepts, so PCR, using micropipettes, calibrating pipettes, how to purify samples of nucleic acids using spin columns, gel electrophoresis, so running agarose and page gels, and characterization using native page. We also discuss a lot of concepts behind transcription and polymerase activity, such as the role of magnesium in both DNA and RNA polymerases to connect chemical reactions. So the catalysis of polymerase for catalyzing polymerization of nucleic acids. And at the end of the day, to actually have a sample of this RNA that they design and to test the purity and folding. So for a little bit of this, so at the end of module 1, students design an RNA with a mutation and they evaluate its predicted secondary structure using the RNA fold web server. And so they end up with something like this. Red means high confidence in the prediction, whether it's paired or unpaired. So this particular construct had high confidence. This was the accurate secondary structure. We also use a primerize, which was developed by the DOS lab to design primers for a primer overlap extension so that students can get some experience using PCR to make their DNA template. All right, so students perform PCR and they run these agarose gels to evaluate that they have the correct size molecular weight band. They also do in vitro transcription to generate their RNA product from that DNA template. So I'm showing here several students. So students are designing their own constructs. So some are bigger, some are smaller. So this is a polyacrylamide gel. Students cast their own gel, they run their samples. Here students are evaluating the impact of the amount of DNA. So students either add like 1 microliter of DNA here or 2 microliters to see if doubling the amount of DNA impacts their yield. We also talk about abortive transcripts and other off products that might take place during the transcription reaction. All right, so for the last module here, we bring it all together. We have several activities that are really centered around high throughput assays. So students learn how to use multi-channel pipettes to prepare 96-well assays. First, they evaluate the impact of metal ions. I don't know if you remember, but there are several magnesium ions that were in that complex 3D structure. So we evaluate the impact of metal ion concentration on that Pepper HBC complex formation using fluorescence as a readout. They compare the binding of their mutant designed RNA to the wild type RNA. And then we also have some computational programming aspects. So students learn some basic Python using Google Colab notebooks to plot and fit their data. All right, so this image was created using BioRender by a really awesome grad student in our group, Emmanuel. And so the basic ideas here is that students are using a 96-well assay and a multi-channel pipette. And so we have a lab just to have some students gain some proficiency using colorimetric assays. And I'm going to show some, this is student data. So in the teaching lab setting, so it may be a little messy, but here a student was evaluating the impact of metal binding. So on the x-axis is a concentration of magnesium chloride, the y-axis is the normalized fluorescence intensity, and so you can see the wild type RNA has a really nice titration curve. This one is a little messier. This is where the amount of HBC is titrated and the fluorescence readout was evaluated. And then student design. So it's really funny to get a window into student ideas. So nearly every student for both iterations of the course they've always tried to design a mutant to improve binding. So I always say, I'm gonna, change this residue. It's going to improve binding, improve fluorescence. But that has actually not borne out in any design. All mutations have weakened binding, some to much more dramatic impacts than others. So one really dramatic impact here was that the students swapped this UG pair with a GA pair, and they found that it abolished binding completely. This other student swapped this AC residues in this loop with a G, believe a C and a G here to close this loop up and form some canonical pairs. And they also saw reduced binding, although it wasn't quite as significant as this UG pair. And so every student has their own design. And so we're growing a list of mutations that impact binding to various degrees. All right, so I want to just spend a couple minutes talking about the impacts on student learning and confidence in these topics. And so at the end of each course, I've administered a survey. And so I just want to kind of preface this with some limitations. This is a pretty small course. We have about 12 students who took the course in 2023 and 7 students who took the course previously to that. And so there's very few respondents on the survey with large deviations. And so definitely take these results with a grain of salt, but I'm pretty excited by some of the preliminary data that we're obtaining here. All right, so this is just one of the questions from the survey. So it's a retrospective survey. And so we asked students that before taking the course, how confident were you in your knowledge of these following content areas? And there are many of them, ranging from topics in each of the modules, including organic synthesis, high throughput assays, performing titrations, et cetera. And so we found that there are really substantial gains in nearly every area, specifically in small molecule characterization. A lot of the students had already had experience in organic labs with mainly GCMS, maybe HPLC, but the breadth of including many different methods really seemed to shore up students' confidence in characterization of small molecules, high throughput assays, RNA folding, and molecular modeling. Where we saw fewer gains was in performing titrations. So looking more closely at the data, students already had a pretty high confidence in performing titrations. They had a lot of experience in prior labs, and so they didn't really report much of an improvement here. So that makes sense. And we're still kind of digging into the data here. Again, the error bars are quite large, but this was really, I think, a promising kind of first insights here. I also wanted to include some comments. They're a little lengthy, but I think that they're really informative. So this question here, in what ways have lab activities affected your critical thinking and problem solving skills? So this student really remarked that there were a lot of concepts that were novel to them. They really had to be exposed to lots of different ideas and do a lot of critical thinking and reading. And integrating lots of different ideas, particularly in the beginning of class about aptamers, folding, and ligand interactions. And so in the past, courses have really been focused on memorization, but this class really forced application. And also where things rarely go to plan. So in this class, because it's really research-based, I essentially invite students into my research group for the semester, and we do lots of experiments focused on things that we already do in our group. And so there's always things that fail that students need to troubleshoot through. So it's really exciting for them. And then for this question, in what ways could the lecture and lab activities apply to their own paths and their own research? So the student found they were extending the critical thinking skills outside of this class to other classes. And then this particular student said that they really needed to restructure the way that they think about chemistry and problem solving, particularly troubleshooting and anticipating problems. So not assuming things are going to work going into it, having an open mind, and developing different types of critical thinking skills. And so with that, I'd like to thank a lot of people. So first, I'd like to thank the outstanding TAs that I've worked with, Glory Romana Rocho and Vishaka Jayasekara, who were really fantastic in helping ensure this course's success. I'd like to thank all the students who've taken the course in the past 2 years. We've had, in spring 2023, we had several undergraduate students take the course, and it's looking like we'll have even more undergrads in the course in the next offering, which is really exciting. I'd also like to thank the group members in my own research group who are just really fantastic and just so amazing to work with. Again, I'll highlight Momodu's talk tomorrow on some of the structural dynamics recent results that we have on the 7SK RNA. And I'd also like to thank NSF for funding this project through the Career Award. And I'm happy to take questions. Thank you.